Do you know what is this? This is the international symbol of female. What is the starting point of formation of the human body? Here is a view of nearly the starting point. We have two gametes. One is a female gamete and the other is a male gamete. They should come together, and once the nuclei of these two structures are met and fused, then we will start having the starting point of creating a new life. Now, this large cell is the ovum. It is the female gamete. Sometimes gametes are called sex cells. And it is a single one. And these sperms are trying to fertilize or to join the nucleus of the ovum, but only one is going to succeed. Now, the ovum and the sperms, they need suitable environment to survive in. Before these gametes develop, we should prepare the place where these gametes are going to live. Now, what is this place or this environment? It is what is called the genital ridge. Genital means reproductive system and the ridge means an elevation. And this is where the story begins. Let us see what this image is telling us. We ask a few questions. What is this structure? This is the future human being. It is the future embryo. Now, this is called the embryonic disc. The human body develops at its first stage as a disc, a flat, oval-shaped disc. Above the embryonic disc, we have a cavity called amniotic cavity. Don't worry about the word amniotic because you are going to hear about it later on as well. This cavity is filled with fluid. Why with fluid? For the sake of nourishment of the embryonic disc. Now, under the embryonic disc, we have another sac, the yellow colored sac, and this is called the yolk sac. What is it filled with? It's a space filled with fluid, nutrient fluid for the embryonic disc. If we follow this arrow, we find that this yolk sac has an extension into the umbilical cord. This is called allantois. We will know shortly why we should know these things. And at the tail end, with the allantois projecting into this structure, at the tail end, this is going to form the umbilical cord, going from the umbilicus of the fetus later on to the placenta in attachment to the mother's uterus so that it gets nourishment later on. This diagram is going to tell us a lot. So let's ask few questions and try to explain. What is this red line? It is the midline between right and left halves of the embryonic disc. Remember, at early stages, the human body is a disc, an oval disc, just like a piece of paper. Now, at later stage, few days later, the one paper disc-like structures is going to be three papers, one on top of each other. The top one, the blue one here, is the ectoderm, the outer layer. Your skin is from ectoderm. And then we have the green colored structure, which is the middle paper of the three, is called mesoderm. Your muscles and other structures under the skin, they develop from this middle layer, the mesoderm. Then we have the lower paper of the three, the lower layer of the embryo is called an endoderm. For example, the lining of your stomach and intestine is developed from this endoderm. Now, this mesoderm, the green zone, is going to be segmented into three areas. This area inside this oval shape is near to the midline, therefore it is called around the midline. The term is para-axial. Axial means the midline and para means around. Then we have this segment between the three segments, and this is called the intermediate mesoderm, and the one to your left is the lateral segment of the mesoderm. We are in the subject of discussing the place where gametes develop, and this place in the middle or intermediate mesoderm, this is the place of development of the place that is going to house the very beginning of formation of the primary sex organs. It is called the genital ridge, and this is the place where it begins, and then the pink layer, the endoderm, is going to send cells into this intermediate mesoderm. This image is telling us few facts about the formation of the genital ridge, the house that the gametes are going to be formed. And let's ask a few questions. What is this structure inside this rectangle? It is the genital ridge. And what is this arrow indicating? It is indicating that the cells of the endoderm are going to invade the mesoderm and start collecting cells on the inside of the genital ridge. These things happen in week 4 of the beginning of fertilization. Now we are going to extract some knowledge out of this image. In week 6, that is after 2 weeks of the beginning of invasion of the epithelial cells of the endoderm into the genital ridge, they are going to divide and proliferate, and their origin is from the coelomic epithelium, the epithelium lining the yolk sac. And what is the arrangement of these invading cells? They are in the form of cords, sex cords. Now, why are they called primary? Because they are the first to develop. Here is another image, and it tells a lot of facts. So let's ask ourselves a few questions. Now, we will start by noticing the genital ridge. Okay? And there is a genital ridge on the right and a genital ridge on the left. You remember the allantois is the little extension of the yolk sac into the umbilical cord, and the very beginning of the sex cells, the primordial germ cells, they start to differentiate and form around the allantois, and then they migrate. These are the primordial germ cells. They migrate. Look at the arrows. They migrate along the nearest structure to them, which is the large beginning of the large intestine. And once they reach the genital ridge, they go into the mesoderm of the embryo and start to settle down in the genital ridge between the primordial cords, on both sides, of course. Here are two figures repeating what we have just said in the previous slide. This is the primordial germ cells in the yolk sac around the allantois, and as they develop, they migrate along the nearest structure, which is the beginning of the large intestine, the third part of the intestine, which is called hind gut, and they move and they enter the posterior abdominal wall made of mesoderm, and they settle down in the genital ridge. Here is the yolk sac, and the primordial germ cells travel along the hind gut, the beginning of the large intestine, and they move around, and once they reach the midline, some will go to the right and some will go to the left and settle down in the genital ridge or the gonadal ridge. Let us check our understanding of the few events that we have just went through, and we were talking about primordial germ cells. First of all, we know that they give rise to the gametes. In the male, they form the sperms, and in the females, they form the ova. And we know for sure now they appear in the wall of the yolk sac near the allantois, and they migrate, they travel along the allantois to the structure attaching the hindgut to the posterior abdominal wall called dorsal mesentery. Don't worry about this dorsal mesentery, you will learn about it several times in the future. And then they reach the gonadal ridge, and we have seen that, and they become associated with the primary sex cords. They live around them. And in the female, these cells become oogonia. They were primordial germ cells. In the female, they become what is called the oogonia. They develop a little step forward, and they divide by mitosis in the genital ridge during fetal life, and no oogonia are formed after birth. So that is a later event. Now, if the primordial germ cells fail to reach or to migrate to the genital ridges, then the ovary does not develop. It is the same story for the testis, the male primary genital organ. If the process is not complete and the primordial germ cells do not reach the genital ridge, the ovary or the testis, the primary sex organs, will not develop. Let's check our understanding again. Now, gonads begin development as what? As the genital ridges, one on the left and one on the right. These ridges, they develop during the 5th week of gestation or intrauterine life, and they come from intermediate mesoderm. Do we know what is intermediate mesoderm? The answer is yes. We have just described it as at one stage the embryonic disc was a single piece of paper, then it developed into 2, and then it developed into 3. The middle paper or the middle layer of the embryonic disc is a mesoderm, and the mesoderm divides into 3 segments, and you can refer back to the slide and see that the mesoderm is going to divide into 3 segments: the paraaxial, the intermediate, and the lateral. Now, there are epithelial cells of what? Lining the yolk sac. What is the yolk sac? The yolk sac is the cavity underneath the embryo, the future human being, and it is filled with nourishing fluid. These epithelial cells are finger-like bands, and these finger-like bands will get into the gonadal ridge, and then these finger-like bands are irregularly shaped primary sex cords. Why irregularly? We have seen the image. They are branching and occupying the genital ridge. And these are the primary sex cords. If there's a primary, then there would be secondary ones. But we are here at the stage of primary sex cords. These cords will grow into the genital ridge, and they form an outer cortex. Any outer layer of anything is called a cortex. And the inner medulla, the inside of structures. And this is the stage of indifferent gonad. What does that mean? It means that all the processes we have just described, they happen in the male and in the female. So the genital ridge now, at this point, is the same in the male and in the female. And it represents the place or the house that the primordial germ cells will come and live in it. After seeing the previous slides telling the story of formation of undifferentiated sex organ, which is the same in the male and the female, now the next short story is the story of developing this undifferentiated sex organ into an ovary in the female. This change from undifferentiated into the primary sex organ of the female, which is the ovary, is going to happen because of what? First, because of the X chromosome in the pair of sex chromosomes, and because of the genes and the hormones. And we will have development of the female genital system, which you can see in the figure to your left. Where is the female genital system? It's in the pelvis, and it opens into the outside through the vagina. Now, the vagina is a fibromuscular tube that connects the cervix to the outside. Now, details of this female reproductive system will be addressed later on when you take the urogenital system. Where is the next arrow pointing? It is pointing to a canal connecting the uterus to the vagina, and it is called the cervix. Then it is the uterus. The uterus is a central part of the female genital system, and it is a pear-shaped structure. The details will be postponed later on, but this uterus is the place of developing the embryo. Now, in this uterus, there is a cavity, and the cavity is lined with special cells called endometrium. This layer is going to receive and do the implantation of the embryo. The wall of the uterus is made of 3 layers. The inside is the endometrium. The muscular part is called the myometrium, and then the outer layer is covered by a special serous membrane called the peritoneum. Then the uterus is going to open into 2 tubes, one on the right and one on the left, and these are called fallopian tubes. Sometimes they are called the uterine tubes. And then we have the ovary. The ovary now is differentiated into a female sex organ, and it is the primary sex organ. Why is the word primary there? It is there because it is the place of developing gametes, developing sex cells. Now, what are the other parts called? The other parts are tubes, different types of tubes for the migration of gametes. Although migration of gametes is an important process, which we will give a summary of it later on. What is this figure? This is a figure of a cross-section of the ovary, the female primary sex organ. Now, what is this image telling me? First of all, it is telling me that the outer layer is the cortex, and in the cortex we have the first, or the primary oogonium, the first stage of starting to develop into a mature ovum, and we will show you examples of this cell. Then the oogonium is going to develop by adding more cells on the outside, and this is the primary follicle, which is going to add more layers, and then it is called multi-layer primary follicle. And when the surrounding cells start to develop cavities, it is going to be secondary follicle, and these cavities will unite with each other and form a large cavity. And once we have a large cavity, then this is called Graphene Follicle. Now, as you can see here, this Graphene Follicle has a big cavity, and the surrounding cells, or the Granulosa Cells, are attaching the ovum to the periphery of the cavity, that's the antrum, and then as it gets larger and larger, it is going to stretch the outer surface of the ovary, and then it ruptures. The follicle ruptures, and the oocyte is released from the ovary. What is this image? You know, pictures talk 1,000 words, and we can extract what we want from this image. What is this structure inside this yellow oval shape? The answer is there are two things. We can see that there is a big cell surrounded by cells, and this is called the Primordial Follicle, the least developed follicle in the ovary. And what is this image? You know, pictures talk 1,000 words, and we can extract what we want from this image. What is this structure inside this yellow oval shape? The answer is there are two things. We can see that there is a big cell surrounded by cells, and this is called the Primordial Follicle, the least developed follicle in the ovary. And the large cell inside this primordial follicle is the oocyte, the primary sex cell. And you can see that this oocyte is surrounded by dark sausage-shaped nuclei, and here it is a single layer, and these are called follicular cells. From where do follicular cells come? They come from the surrounding tissue to the primary follicle. The questions and answers opened our mind to notice lots of things. In week 10, this is what is going to happen. In week 10, we will find that the genital ridge is undifferentiated gonad. It is the same in the male and in the female. Then, because of several factors we have just mentioned, this undifferentiated gonad is going to go a step forward and becomes identifiable ovary because of the genetic of the X chromosome, because of hormones, and the primary sex cords, which we talked about, and they are the epithelial cells of the oocyte going into the genital ridge, and we talked why we call them primary, because they are going to change in nature. These primary sex cords will degenerate, they will die and disappear, and secondary sex cords are going to appear, and they are called now cortical cords, because they will not grow all the way, they only grow into the outer layer, and that's why they are called cortical. And these cortical cords, they come from the surface epithelium, the same origin of the primary sex cords, and they grow into the genital ridge, which is made of the mesoderm, the intermediate mesoderm, and the word mesenchyme is used as another term for the mesoderm. And when these secondary sex cords enter the genital ridge, they find that there are primordial oogonium, primordial female sex cells, while the primary sex cords, when they went in, it was only mesoderm. So the situation now is different, and the entry of these cortical cords, they are there to form a follicle which is called primordial follicle. So the primordial follicle, which is diploid in number, diploid is a term describing the number of chromosomes, and in diploid cells it is 2n. That means it contains the whole set of 46 chromosomes. These primordial follicles, they are made of an oogonium, the very early female sex cells, plus a single layer of squamous follicular cells. We know a single layer is one layer, a layer made of one group of cells, and squamous means thin, and the follicular cells are the cells of the secondary sex cords that went in and divide in number and fill the space. And these cells making the single layer around the oogonium will start with the help of the oogonium to form a layer, a glycoprotein layer surrounding the oogonium and separating it from direct contact with the follicular cells, and this is called zona pellucida. Zona pellucida has a story, and we will be talking about it later on. What is this view? It is a true histological section of the cortex of the ovary. What can I see in this section? If you notice the follicle inside this yellow circle, you can find a clear cytoplasm, whitish area, and the nucleus is in the middle. This is a very early oval cell, and it is surrounded by a single layer of thin cells. Thin cells are called squamous cells. If you ask yourself how many layers are there, the answer is a single layer. And here is another example in a nearby follicle containing an ovum and surrounded by a single layer of thin cells. You know it is a single layer because you can see these dark spots which represent the nuclei of the surrounding cells. These are features of what is called primordial follicles. These are the very early stages of development. When we go around and look for other follicles, we can see a follicle here. First of all, the ovum or the oocyte is larger, and we can see that the surrounding cells are not thin because the nuclei are not like a simple line. Nuclei of the surrounding cells are cuboidal, like a cube, and these are features of the next stage of developing primordial follicle, and now it is a primary follicle. Then you compare between an ovum surrounded by a single layer of thin cells and an ovum at the tip of the curved arrow. You can see that there is a difference between the single layer lining of a primary follicle and a single layer flat cells surrounding the oogonium. This is another histological section of the cortex of the ovary, and when we are curious, the questions will help us extract information. If we look at the large follicle in the middle, and we can see the oocyte, the primary oocyte. Now, why is this primary oocyte, and why is it a primary? Because if we can see, this yellow structure is pointing at that the surrounding cells are cuboidal. They are not flat. Cuboidal means that the two dimensions of a cell are about equal, and this is the description of cuboidal cells. Then we go to the point of this arrow, and we can see a dark line surrounding the ovum, and this is glycoprotein material secreted by both the ovum and the follicular cells, and it is called zona pellucida. Now, for this reasons, this is a primary follicle containing primary oocyte surrounded by single layer of cuboidal follicular cells, and the zona pellucida has appeared. Let us check our understanding of a primary follicle and why it has changed its name from primordial to primary. We have noticed essential basic facts, which are that the oocyte is getting larger and it starts to have more than one layer of follicular cells. What is next is that these follicular cells, they become to be known as granulosa cells. Why granulosa? Because they are starting to develop granules of hormones to be secreted. Now, these two layers become more than two, and they are multiple, and then the follicle is to be called multi-layered primary follicle. Now, the oocyte itself is arrested at a very early stage of first meiotic division, and that is the prophase. And do you know when is it going to complete this? It is going to complete this first meiosis after or beginning of puberty. So it is going to stay in this place, in this phase of meiosis for 12-13 years, and therefore it is exposed to chances of being changed. Then the zona pellucida. Zona pellucida is a thick glycoprotein layer that is situated between the oocyte and the zona glomerulosa, made of granulosa cells, and it is in a stage of meiosis 1. This is a section of the ovary in which we can see what? We can see that the granulosa cells are now multi-layers, and we can see that it is called multi-layered primary follicle. Now, what is the principle of this slide? This slide is for the sake of comparison. Comparison between what? Here we can see an oocyte surrounded by zona pellucida, and the granulosa cells are multi-layered with large nuclei, and these are granulosa cells coming from the surrounding tissues of the oocyte, and therefore this is multi-layered primary follicle. While if you go up, you can see that there is a primordial follicle where it is having smaller oocyte and there is no zona pellucida, and it is surrounded by a single layer of flat cells. Let's ask questions about this section. First of all, the story of embryology is that it is made of sequence of events at specific times. Now, in this section, what kind of follicle is it? It is multi-layered primary follicle. But if we look at the tip of the yellow arrow, you can see that there is an attempt of forming a small space. This small space is going to change the name of this follicle. But this space is very small, and any space is called an antrum, and this is a small antrum. Therefore, this development is going to change the name of this follicle from primary follicle into secondary follicle. With the help of curiosity and asking questions, now we have a follicle in the center of this slide. Now, the little space we have just seen in the previous slide is getting larger and larger. And in addition to that, more spaces are developing in zona glomerulosa. And what do you think is going to happen next? This is a secondary follicle because of presence of vacuoles containing fluid in zona glomerulosa. Now let us collect some basic important facts about a secondary follicle. It is very similar to primary follicle, meaning that large oocyte and multiple layers of zona granulosa. Then they are larger. Secondary follicles are larger than primary, and they start to have more follicular cells. Follicular cells, you know, they have another name, and it is granulosa cells. There is going to be small vacuole production, and these small accumulations of fluid in the intercellular spaces of the granulosa cells. These spaces, gradually, they are going to join together, meaning to coalesce to form a large cavity, and this cavity is called an antrum. Now, there are also surrounding granulosa cells, and these cells surrounding the oocyte are going to still connecting the oocyte to the granulosa cells. And this connection between the oocyte and the granulosa cells is called cumulus oophorus. Then things will develop. Now, the surrounding granulosa cells are now to be called theca. And there are two layers of the theca cells. The theca interna is a group of granulosa cells surrounding the Graphene follicle, the follicle that has a single antrum, and the theca externa on the outside, and it contains more fibrous tissue than cells, and the theca interna is going to secrete enzymes. Therefore, if we go back and remember that we called the follicular cells, which are growing in numbers surrounding the oocyte, they are going to start producing granules. Therefore, their name has been changed from follicular cells to granulosa cells. We need to ask questions. What is this section? Here we can see that the little spaces in zona glomerulosa has united together and formed a large single cavity filled with fluid. These cavities filled with fluid are important for the nourishment of the oocyte. So this is the last stage of developing follicles. This black arrow is indicating the oocyte, and this arrow below it is pointing at the granulosa cells that connect the ova or the oocyte to the granulosa cells, making the periphery of the antrum. This number of granulosa cells are called cumulus. What is a cumulus? It is simply granulosa cells of theca interna connecting the oocyte to the granulosa cells. And these are the features of the last stage of development of the oocyte with its follicles, and this is the Graphene Follicle. You know something, when you learn pieces of information and when you take a pen and a piece of paper and write what you have learned, this is going to be a very useful way of learning. We have just seen the Graphene follicle, and we want to write down our observations. First of all, there is a single space. This single space is called the antrum. It is filled with fluid for the sake of nourishment of the oocyte. This antrum is surrounded by what is called membrana granulosa. It's a cover or structure surrounding the antrum and the oocyte, and these are granulosa cells. These granulosa cells surrounding the antrum and the oocyte, they project into the antrum, and this is called cumulus oophorus, which we have just seen, and they connect the oocyte to the surrounding granulosa cells. Now this Graphene follicle is getting larger and larger. Why is it getting larger? Because of more fluid is accumulating in the antrum. It reaches the surface of the ovary, and when it increases in size more than the cover of the ovary can stand, the Graphene follicle will burst, and the oocyte is expelled outside the ovary. This is ovulation. What comes out of the Graphene follicle is the oocyte, the zona pellucida surrounding it, plus the group of Granulosa cells that are surrounding the oocyte. So there are three structures expelled at the point of fertilization. The corona radiata, they are no more than follicular cells. At fertilization, meiosis II is being completed. We need the aid and the help of questions. What is this diagram? This is the diagram of the expelled oocyte with its coverings. And let's review what are the structures surrounding an oocyte. It is corona radiata. These are granulosa cells on the outside, and then zona pellucida. And on the inside of zona pellucida, there is a thin layer of jelly material called vitelline membrane. And then we have the oocyte, cytoplasm, and the nucleus. Some people call the nucleus of the oocyte as germinal vesicles because it is going to divide and divide, and the repeated division is described as germinal, and the nucleus, the very important, now it is 1n, half the number of chromosomes. And with the aid of questions, number one, have we seen this section before? The answer is yes, we have. It is what? It is a diagram of a cross-section of the ovary, the primary sex organ in the female. At that time, we were discussing the types of follicles, and in the last few slides, we reached the stage of the oocyte being expelled. When these events are happening in the cortex, and at this event, when the oocyte is expelled, what remains in the cortex is the granulosa cells. These are hormone secreting cells, and they have granules. That's why they are called granulosa cells. These cells, they start to secrete hormones. In fact, they have started secreting hormones much earlier than this event, and it becomes functional, and this is what is called developing corpus luteum. This is what? This is what remains of the granulosa cells. And when it develops, it is well-established structure, and it is yellow in color, and this is the functional, the fully functional granulosa cells, and it is called corpus luteum. If pregnancy is happening, this corpus luteum is going to stay active and secreting estrogen and progesterone, promoting pregnancy. If pregnancy or fertilization does not happen, there is going to be no need for these hormones, and no need for the granulosa cells, and no need for the corpus luteum. Therefore, it is going to atrophy. That means it stops functioning, and when structures stop functioning, they get smaller and smaller, and this is the last non-functional corpus luteum, and it is called a scar, corpus albicans. It is the degenerating functional corpus luteum. You know something, it is always useful to write down things, because when you write them, first of all, you test your short-term memory. Secondly, it elevates your level of understanding. And let's have some very basic facts about the corpus luteum. What is the color of the corpus luteum? It is a yellow color. How do I describe it? It is a glandular structure. What is it made of? It is made of active follicular cells. What is another name of the follicular cells? It is granulosa cells. Where is corpus luteum? It is in the cortex of the ovary. When does it form? It is formed when the Graphene Follicle ruptures and discharges the oocyte. If it is a glandular mass, what does it secrete? It secretes large quantities of what? Of pregnancy promoting hormones, hormones that act to make pregnancy continues. And what are these hormones? They are the estrogens and progesterones. Is it going to stay active and secreting? The answer is no, it depends. But when pregnancy does not happen, it is going to be atrophied. That is, it shrinks and degenerates into a white scar in the cortex of the ovary called corpus albicans. If pregnancy occurs, what is the situation? The corpus luteum, in case of positive pregnancy, it will continue secreting the pregnancy promoting hormones until parturition. What is parturition? Parturition is the process of giving birth to a baby. This is a group of laparoscopic photographs. What is laparoscopy? It is putting an instrument into the abdomen through an incision and shining a light and watching the structures live. And in this image, we can see the red bulging area is the very distended about to rupture Graphene follicle, and the second image to your left, you can see that there is an oval yellowish structure protruding from a point. This is the oocyte coming out of the Graphene Follicle, and in this image the oocyte is about to be separating from the Graphene Follicle, and the lower right image is about the ovum or the oocyte is to be falling down from the ovary. This is a good practice for you to comment on, and I will help you during this process. Which structure is the whitish large structure in the image, and what is this bulging reddish coming out of the surface of the ovary, and here is what is going to be expelled out. What is this structure? It is the oocyte about to drop out of the Graphene Follicle. I have put this slide for you to reactivate your knowledge of the different parts of the female genital system. Now, if you ask yourself questions and try to identify the following structures. What is this structure? You know this structure after the previous few slides. And what is the name of this structure? And these arms are coming from the last part of the fallopian tube or the uterine tube, trying to surround the ovary so that when the ovum is thrown out, these fimbria will catch the ovum and take it in. After the fimbria, there is this structure of the fallopian tube, and it is called the ampulla. In this place, the ampulla, fertilization will take place. Now, you may ask, fertilization is fusion of the oocyte with the sperms. How come that the sperms reach this area? This is going to be explained later. This is a diagram of the uterus to your right, and you can see the ovary, and in this area, inside this triangle, you can see that the Graphene follicle has ruptured and the ova or the oocyte is dropping down. This is a process that is very important because it has to be caught by the fimbria of the fallopian tube. If the fimbria of the fallopian tube are not functioning well, then this oocyte is going to drop down in the pelvis. And soon after the fimbria catch the ruptured Graphene follicle and catch the oocyte, the sperms will be arriving and fertilization will take place. What is this image? This is a three-dimensional image of the lining, the inside membrane of the fallopian tube, And you can see that when the oocyte or the egg or the ovum enters the fallopian tube, these extensions of the lining of the fallopian tube, they are called cilia, and they move, beating things from the ovary end of the fallopian tube to the uterus. Here is a diagram of what happens in cases of development of the oocyte. It starts as oogonium, and it has full set of chromosomes, then gets larger and surrounded by follicular cells. It is called primary oocyte. This primary oocyte, which is 2N, having full set of chromosomes, meiosis 1 is going to start here, and meiosis 1 is going to stop at prophase. And then when reaching puberty, the primary oocyte will change into secondary oocyte by changing the shape of the follicular cells, and it is trying to become 1N, and here meiosis 2 is going to be completed at puberty before fertilization. What is this? This is a mind map. What is a mind map? A mind map is the most powerful thinking tool present in the 21st century. What does it do? It presents the key words, basic facts, and integrate them in their right place in an A4 piece of paper. Now, it is the best way to test yourself. If you can mind map it, you know it. If you cannot mind map it, you don't know it. When you don't know it, you go and learn it. So it is a very good way of telling you the truth. You know this and you don't know this. Therefore, you go and remedy your knowledge. This is a mind map of oogenesis, which is the process of formation of oocyte. Where does it start? It starts its story in the yolk sac, and we have went through this information, but we are summarizing it and putting things in its right place. That is integration. What do we know about the yolk sac? It is an extra embryonic cavity inferior to the embryonic disc, which is the future human being, and in the yolk sac there is production of primordial germ cells. The next idea is that there is a structure called Genital Ridge, which is a very early ovary. It is the skeleton of the ovary. What is the basic knowledge of the genital ridge? The genital ridge is a shelf-like structure medial to the future kidney, and it is higher up in the posterior abdominal wall, and it is going to house the primordial germ cells, which are called oogonia. These cells, the oogonia, they are going to multiply immensely by mitosis, and some cells will get larger. And when these cells get larger, they are surrounded by a group of cells called the follicular cells, which they come from the ovary surface. When an oogonia is surrounded by follicular cells, it is a primary oocyte. This primary oocyte is going to start meiosis 1, but stops at prophase and continues after puberty. Then there is more mitosis until a huge number is achieved, approximately 7 million primary oocytes present in each ovary. Now, this is going to stop, and primary oocytes are going to die, large number of them, and this death is programmed in the genes, and it is called apoptosis. What is apoptosis? It is programmed death of cells. Then we come to 5 months while the embryo is inside the womb. During this age, most of the primary oocytes are dead, and by the 7th month, the number is reduced, and these structures are near the surface of the ovary. These ones are going to survive, and these are primary oocytes that remain alive, surrounded by follicular cells, single layer of follicular cells, and they are called primordial follicles. And by the 7th month, there are about 2 million. After birth, everything is going to stop and rest. This stage is called diploitin, stage of standstill. After birth, there will be the childhood, and during the childhood, more death of the primary oocytes and the oogonia. And these primordial follicles are going to be sacrificed more and more. Then comes puberty. At the age of, the common age of 12 or 13, sometimes earlier, there will be the stimulus of hormones. To what? To the follicles. What hormone? The hormone is the follicular stimulating hormone, a very descriptive term of what this hormone does. And when these follicles are stimulated, 5 to 15 of the primary follicles are developed at a time. And at this stage, at puberty, there are around 400,000 primary follicles. Then, when the primary follicles receive the stimulus, these primary follicles contain primary oocyte and follicular cells around them, several layers of the follicular cells, and they get larger. Then comes the secondary follicle, which is larger than the primary, and the secondary is when the granulosa cells or the follicular cells develop cavities, and these a single cavity is called an antrum, and then they unite to form one large space which is filled with fluid, and there is the formation of zona pellucida, which started at the primary follicle, and it continues the process of formation of the zona pellucida. Now we're finished with the secondary follicles, and we come to the Graphene Follicles. These are called Graphene Follicles because they get larger, more fluid in the antrum, and it is about to ovulate, about to get out of the follicle. These cells, the Graphene Follicles, are going to resume meiosis 1, which was stopped at the early prophase many years ago. And when meiosis 1 resumes and meiosis 2 starts, the division is going to result in one daughter cell and a polar body. What is a polar body? A polar body is half of the chromosomes with little cytoplasm thrown away. So the 2N cells at this stage, they become 1N. And the Graphene follicle is going to start meiosis 2 and also stops at anaphase, and it is ovulated at stage of prophase of meiosis 2. Once it is ovulated and thrown out, the oocyte dies within 24 hours.